Hello, welcome to Summertime Science 2021. My name is Rodrigo Vega. I'm a senior lecturer in biology at the section of Natural and Applied Sciences at Canterbury Christchurch University. Today I wanted to tell you about the molecular ecology research that we do at CCCU. And I wanted to tell you about one specific example. I want to tell you about the case of peregrine falcons, which is a formidable animal, and I wanted to tell you about the research that we have been doing on this group of animals. So you might know that ecology is the study of the abundance and distribution of organisms and the interaction with the environment. So what is molecular ecology? In this case, molecular ecology is a scientific discipline that studies ecological patterns and processes using or applying molecular biology tools. It's an integrative discipline, right? Bringing concepts and approaches from very different areas. So of course we have to bring approaches from traditional ecology in order to go and collect our samples in the field and collect all the data from the field that we can analyze alongside the molecular data that we might collect from the individuals. But we also bring concepts from conservation biology, from molecular biology, specifically the molecular biology tools that we are applying on our samples, concepts from genetics and genomics, evolution, and biogeography, and depending on the type of questions, molecular ecology might go towards the area of systematics and taxonomy as well. So you can see that we are bringing approaches from the field, laboratory techniques, and computer science and statistics as well to understand the molecular ecology of a group of interest. That's why I see this as an integrated discipline and quite a broad discipline as well. In my case, I am a molecular ecologist interested in studying the evolutionary history of different group of organisms, study their phylogeography or the genetic distribution of the genealogical lineages, their conservation. Sometimes if the group of organisms are working, have any conservation interest, then I try to focus our questions towards the conservation biology of the different species. And I have been fortunate enough to work with different groups of Mexican rodents, and I have an ongoing project on black and white rough lemurs. I had a PhD student working on western lowland gorillas. I did my PhD in pygmy shrews, and the other groups that I'm also working with, specifically DNA barcoding of chironomids or non-biting midges. But today I want to focus the attention to the peregrine falcon. So this majestic animal that, if you're fortunate enough, you might have seen it flying around your city. Now, the question we wanted to ask in our research study is, where do peregrines come from? So you might know that the peregrine falcon has been affected by persecution, by pollution, problems with trade. It's a charismatic animal, and people want to keep it as well for falconry. So it has been extracted from the wild to breed in captivity as well. And obviously from habitat degradation, the loss of habitat for these animals, especially for the habitat where these animals might want to breed and establish their nests. So in many places around the world, we have seen that the population have crashed. However, the peregrine falcon is also a flagship conservation success story because there have been successful reintroductions back into the wild. However, in the UK, and specifically in Sussex, where there used to be a very large population of peregrine falcons, there were never formal reintroduction programs. So this warrants the questions: where do the established peregrine falcons in southern England come from? And if they have suffered population decline, have they suffered those detrimental impacts from a population bottleneck and a genetic bottleneck? So those are questions that we establish and we ask in a research paper where the author is Angela Weaving, published in 2021 in Conservation Genetics. and also working in collaboration with the Sussex peregrine study and others. So I ask you to go and find that paper if you can, as well, if you want to read more about it. So here in this graph, I just want to show you that the population of peregrine in southern England was not observed for about 30 years. So we have very good data before the population crash. Actually, peregrine falcons have been seen in the area for a very long time and recorded since the early 20th century. Then we have World War II, but the peregrine falcons, although they have been persecuted, they were still there. But at some point after World War II, we started using organochlorine pesticides quite intensively. And this is believed to have brought the peregrine falcon into steep decline. And around 1955, 1958, we don't see any breeding pairs anymore for 30 years, and that's from Sussex. Once the organochlorine pesticides had been banned, or they were more restricted in terms of their use, we see a steady increase in the number of individuals that we see again in Sussex. Actually, the numbers that we see today are even higher than those that we have historically. So in Sussex and in other places, we can ask this question, where do they come from? So maybe they were never really gone. Maybe we didn't really look carefully enough and we have missed that there were a few animals here or there. But in Sussex, we're pretty much sure they were not around for those 30 years. It could have reestablished from some animals that escaped from captivity. Sometimes the falconers might lose one of those peregrines, and those peregrines become lost, and they might have established a colony. That's a fair question to ask, right? They could have come from domestics that escaped. They might also come from the continent. They might come just from France. It's just on the other side of the coast of Sussex. Or maybe they come from a little bit farther away. Maybe they come from Ireland, or from Wales, or from Scotland, or from Scandinavia, from what we call refugia areas where the peregrine falcons were not that affected by the organochlorine pesticides and persecution and loss of habitat. So to answer these questions, what we do is we take a conservation genetics approach to study this. So we collected samples of peregrine falcons that are of individuals or specimens that are present in museums. So we have a representative pre-population crash from the UK. And then we collected more fresher samples, a post-population crash in the UK. We also collected samples from domestic, from Irish and from German populations, other continental samples as well, and data that has been collected by other research teams and deposited on gene banks, so DNA sequence data. Then we're looking at two different types of markers. One is a maternally inherited marker known as a mitochondrial DNA control region. It's a region within the mitochondrial DNA that is known to be hypervariable, that we can use it to see how many variants of mitochondrial DNA control region we have within a population or populations. Then we also looked at nuclear markers. So those are inherited from the paternal line and the maternal line. And we decided to use 7 different loci, locations in the chromosomes, with strands of DNA that are known as microsatellites. That are also hypervariable, and individuals can carry different alleles within a population, right? So we're looking at the haplotype variants that we have in control region on one hand, and on the other hand, we're looking at the different genetic variants or alleles that we have in nuclear DNA. And once we have collected this data, then we start looking at, okay, how genetically diverse our populations are in terms of mitochondrial DNA and in terms of nuclear markers. And we ask the question, okay, how many heterozygotes we have, and how many haplotypes do we have? And then the other question we ask is, okay, now that we have this variation, this genetic diversity, how is this genetic diversity structured across the populations? Do we see that individuals from different populations share those variants, or do we have populations that do not share those variants? So this is talking about the genetic structure of the population. So these are typical approaches that we use in phylogeography and population genetics. We collected the data, we analyzed it, and we saw that overall we have low genetic diversity for the UK populations before and after the population crash. So this is highlighted here by the number of alleles and the heterozygosity values. And overall, they were slightly lower than the genetic diversity values that we saw in the Irish and domestic groups, which have a few more alleles, and overall the heterozygosity values were slightly higher than those in the UK population. And this is mirror also between microsatellites and mitochondrial DNA. So we see a pattern here of UK peregrines being less diverse than those from Ireland and other groups, and the domestic groups as well. Now that was in terms of the genetic diversity, but now we want to know how is this diversity structured in the populations. And in our analysis, we discovered 4 clusters or groups. One is the UK post and UK pre, in this case, the same color of green. So genetically, those 2 groups are very similar and they share the genetic variants. The domestic group is genetically different from all the others, as well as the German group is genetically different from all the others, and the Irish group is also genetically different from all the others. So that's why we are talking about 4 different clusters, distinguishable based on microsatellite data. In terms of mitochondrial DNA, the mitochondrial DNA is not as variable in falcons in general. And we observe very few variants, haplotypes, and we discovered a couple of haplotypes that were preferentially found in the UK post group and the UK pre before the population crash population. So it could be that the UK population before the crash have some variants, haplotypes that were not present, that we don't see anymore now in current times. But overall, there's very little information that we can gather from this mitochondrial DNA. But overall, we have some good information and we can reach some conclusions. So where do peregrines come from? Well, peregrines were definitely gone, extirpated from southern England for about 3 decades, okay? So we know that they were not there already. So there has been an unassisted recovery of peregrine falcons in southern England. However, we have not detected a signature of genetic bottleneck. A little changes in the level of genetic diversity through time. Remember, the UK pre-population bottleneck and post-population bottleneck were genetically pretty much the same. And in terms of genetic structure, were also very similar. So they seem to have been historically having low genetic diversity. So we also know that they do not come from domestics and they do not come from continental Europe. Genetically, they are different from those groups. If they were similar, then we would have seen a genetic signature that puts the UK post population clustering together with the German or with the Irish. So could they come from refugia, from other areas? Could they come from other places in Ireland that we haven't sampled, or from Scandinavian peninsula, or from Wales or from Scotland? This is still a possibility. We don't have data from there, so we need more samples to potentially sample the source populations. Could they come from France as well? It's still a possibility, but from the moment we know that they do not come from domestic animals that have escaped into the wild, they do not come from continental Europe, at least not from the German stock or from other places farther south. And they don't come from the region from Ireland that we sample. So there's still some work that we can do, and for this we need extra sample from other areas, but at least we have done some work towards the understanding of where do peregrines come from. Well, I hope you have enjoyed this presentation. I know we don't have a definite answer, but that's something that typically happens in science. You ask one question and then you try to answer that, and then this brings a different question, or you don't reach the answer, so you keep going and advancing your science. But that means that you as scientists still have work to do. I hope you appreciate this talk. This is Summertime Science 2021, and thank you for watching.